Professor DiCarlo received his bachelor's in bioengineering from UC Berkeley in 2002, PhD in bioengineering from UC Berkeley and UCSF in 2006. After a postdoctoral study at Harvard Medical School, he joined the faculty of UCLA and he's been there since 2008. He is the vice chair of the Department of Bioengineering and a deputy director of an NSF Engineering Research Center focused on diagnostics and underserved populations. His research pioneered the use of inertial fluid dynamic effects for the control, separation, and analysis of cells and microfluidic devices. And he has founded many companies commercializing the IP developed in his lab, including Cytovale, Vortex Biosciences, Tempo Therapeutics, Foresight, Ferrologics, HANA Diagnostics, and Partelian Bioscience. He received many awards, including the Presidential Early Career Award for Science and Engineers. He is elected a Fellow of American Institute for Biomedical and Biological Engineering, and he is a Fellow of Royal Society of Chemistry. He was also awarded NSF Early Career Award, ONR Young Investigator Award, DARPA Young Faculty Award, NIH Director's New Innovator Award, and Coulter Translational Research Award. With that, the floor is yours, Professor DiCarlo.
Thanks, Mehmet. That was a very long introduction. Appreciate that. So as Mehmet, it's really a pleasure. Thanks for inviting me. It's a pleasure to speak. This is a very nice format that I get to talk to people all over the country and all over the world. So, of course, if there are questions, feel free to ask those. As Mehmet mentioned, my background is in the microfluidics area, and I've spent a lot of time in that area developing technologies, commercializing those technologies, and I've learned a lot in that time about what goes well and what doesn't go well in commercializing those technologies. And generally, microfluidics has been used to accelerate biological research and diagnostics. And one of the challenges that has arisen with commercializing microfluidics is that it often requires new infrastructure, new instruments to run microfluidic chips. And that has been a bottleneck in terms of bringing new technologies out there and really scaling the way that we do biological research. So what I'm going to talk about today is, understanding this and kind of learning from my mistakes in commercializing microfluidic systems. What's next? What's next after microfluidics, and what are we doing at the at my lab here at UCLA in order to scale biological research in a way that we think can be adopted much more rapidly and can spread to research labs, clinical labs at a pace that far exceeds what microfluidic technology has. So I'm going to talk about this lab on a particle technology and really want to start off by kind of covering the broad areas of therapeutics that we're seeing now, and how microfluidic lab on a particle technology, how automation of life science research plays a role in really driving these therapeutic discoveries, developing new therapies, manufacturing those therapies, and making and analyzing the products that become therapies in ways that are quantitative and scaled. So the way I think about medicine is we really now have three pillars of medicine. So molecular therapies, where we're really interacting at the scale of molecules in the body. This started with really the small molecule pharmaceuticals. We have RNA-based therapies, and then more recently, in the last few decades, using recombinant proteins or antibodies, in particular antibodies, monoclonal antibodies, as therapeutics. And now 6 out of 10 of the blockbuster drugs that are sold now are based on monoclonal antibody therapies. And then gene therapies, we've seen this revolution in our ability to edit genomes using things like CRISPR or related genome editing approaches, and in using viral delivery approaches. And so gene therapies, of course, are really taking off quite a bit recently. And then cell therapies. Of course, some cell therapies are genetically modified, but others are not. But cell therapies is kind of this third pillar. We've seen amazing successes with chimeric antigen receptor T cell therapies and hematologic malignancies. And we're seeing, a huge investment in that space and development of new types of cell therapies. Beyond engineered immunotherapies, we know that cells secrete things that are really important for their therapeutic effects: growth factors, cytokines, extracellular vesicles. And of course, vaccines. We're all very familiar with vaccines. This has really been a triumph of medicine over the last century. And we've seen how rapidly now we can develop vaccines, particularly using RNA-based approaches. But I think one of the key things in really driving these therapies forward and what has driven them in the past is the ability to screen many single cells. So an example is when you're developing monoclonal antibody therapies, identifying hybridoma cells that are secreting antibodies of interest, or identifying B cells directly, plasma B cells or plasmablasts that are secreting antibodies that neutralize or have some effect. That's a key bottleneck in the discovery of new therapeutics. I'll give another example in gene therapies: the secretion of finding cell lines that secrete lots of virus. Viruses actually becomes a bottleneck in gene and cell therapies, producing GMP grade virus. So finding cells that produce and secrete a lot of virus becomes now a really important task. And then in cell therapies, we have T cell therapies or CAR T cell therapies, where the function of those cells is now based on the ability to secrete things like perforin or these pore-forming compounds, gramicidin, or cytokines that attract other immune cells. And the same, we know now for therapies that are based on mesenchymal stem cells, that a lot of their function has been based on the things that they secrete, the growth factors that they secrete, or the extracellular vesicles that they secrete. And then others are using extracellular vesicles themselves as the product. And so in all these cases, there's some aspect of what the cell is producing. Its function is really connected to what it's producing and putting out and communicating with other cells. And of course, with vaccines, we know that one of the key aspects of how vaccines function is to generate humoral immunity and B cells that are producing antibodies, along with T cells as well. But understanding the repertoire of antibodies that are being produced, that's critical. And also, I think the point here is that to scale the discovery efforts, to scale the understanding of these new types of therapeutics that we're developing, we need to analyze cells, analyze usually single cells, and analyze the functions of those cells, like their secretions and how they interact with other cells. So that's really the challenge that's laid before us, and that's what I'm going to talk about, how the lab on a particle technology can have an impact. And I guess the key here, how do you connect this need with how we do biology today? So what is the vessels in which we do biology? Well, we start, bioreactors at the really large scale, petri dishes, but at the scale that we're scaling biology at its most, it's really automation with robotics and multi-well plates, right? So down to 96, 384, 1536 well plates. And so that's the scale. Of course, that scale is not at the scale of single cells. So single cells are orders of magnitude smaller in their volumes and length scales. And so that's been one of the issues, is if we want to analyze single cells and things that are released from single cells or secretions from single cells, if we're doing that in a multi-well plate that's tens of thousands to hundreds of thousands of times larger volume, then we need to grow up cells to large quantities in order to get signal from things that are secreted, or that there's time scale differences, or we just can't look at the precision needed at single cells in those large volumes because things get diluted out. And so that's where a lot of the microfluidics tools that have tried to address this problem, focus on this. How do you make small volumes at the scale of biology? And so the ways that have been proposed in the past, using microwell plates, like microwells in a surface that are 50 to 100 microns in size, or using droplets themselves. That's kind of the approaches that have been laid out. Well, and I mentioned this at the beginning, the challenge with those approaches is that, making a chip is great, but operating that chip that makes droplets requires a new piece of new instrument. And still, those droplets don't have the solid phases that a well plate does that allow you to do other important types of reactions where you're capturing things that are secreted or capturing things that are released from cells. And so that requires new types of innovations. And so what we've been doing is working on a new type of concept, which we call lab on a particle. And the idea is that very much like a well plate, it has a solid phase. It has a structure to it in which cells can go into that structure. And that structure can be used to capture things that the cells are doing, like their secretions or nucleic acids that are released from those cells. And that structure also can be used to template the formation of volumes that are at the scale of cells. And so this is this idea of lab on the particle. And the key thing is that these structures, these particles, that they are compatible and can be used with infrastructure that's already out there in folks' labs. So things like flow cytometers or sequencers, et cetera, single cell sequencing instruments, et cetera. So that's the challenge, and that's how we're addressing the challenge. So let me go into a little bit of kind of the scale here. Why are we able to do 1,000-fold, accelerate biology? We think 1,000-fold. So think about a 1536 well plate. This is really the ultimate scale in which biology is done. In most cases, there's 1 to 10 microliters in each of these wells. We call these tropicals, or particles that template droplets. Each of those is holding 1,000-fold smaller volume. So really, we're getting very close to the volume of a cell, less than a nanoliter. That means you can concentrate things that cells secrete 1,000-fold. And we can measure 1,000 times more compartments. So we can measure, hundreds of thousands of compartments using the high throughput instruments that are already in labs, like flow cytometers, that can measure hundreds to thousands of events per second. And because they're compatible with cytometers, you can also sort. So sorting and picking a particular colony here requires a robotic instrument or something like that. Here you already have that present in the cytometer system. Okay, so that's the concept here. Just to get into more details, this lab on a particle, I'll talk about a few different implementations, but I'll focus mostly on one type. So you can imagine these as basically sortable nanowells, right? So that they hold nanoliter volumes. They're we call them nanovials. Cells can get seeded onto these particles into their cavities. The cells secrete. We can encapsulate those particles, so we can mix them with an oil so that a droplet forms around this, and that prevents crosstalk. We call these particles that have a droplet around them a dropical, a drop particle, or a dropical. And so that enables you to segment and prevent crosstalk between these chambers. We have different types of lab on a particle technologies that we've developed. One technology we call pico shells, which is a completely enclosed particle in which you can grow colonies. This technology requires the use of microfluidics by the end user who's using it. So this is one type of lab on a particle platform. We have another type where we have different types of particles that have controlled hydrophilicity and hydrophobicity. These will spontaneously form droplets, and we've been using this in diagnostic applications mostly because you can also barcode shapes. But I'm mostly going to talk about this nanovial technology. And so, here's actually a 3D printed version of a nanovial. You can see it's like a bowl, a little bowl here with a cavity inside that cells can fit in. Obviously, this is not to scale. Okay, so I'm going to talk mostly about nanovials. As I mentioned, these are sub-nanoliter containers for single cell analysis. So the volume in the cavity is less than a nanoliter. So the volume of a particle, depending on the size, is about a nanoliter. So that means if you have 10 to the 6 of these particles, that's about a milliliter of particles. So you can hold 1 million of these wells in about a milliliter. The nanovials are functionalized to interact with cells and to interact with things that they do. So their secretions is a good example. So the nanovials are made out of hydrogels in most cases. And so in that case, we can add chemistries to the hydrogels so that cells adhere, and I'll talk more about this, and chemistry so that we can capture their secreted molecules from the cells. In this case, I'm showing an antibody that the cis cell is secreting and being captured on the surface of the nanovial. The nanovials can be operated with pipettes and centrifuges and normal reagents, normal instruments that folks use. So you can exchange reagents around them. You can do wash steps. You can, as I mentioned, seal and unseal the cavities by emulsifying in an oil phase and then breaking that emulsion to bring them back into a water phase. So you can do multiple steps. This is something that microfluidic droplet-based approaches have a difficult time doing, because once you create that droplet, it's hard to add things or remove things from that droplet. This lab on a particle technology is a very simple process that you can do with centrifugation and wash steps. And as I mentioned, these particles can be sorted with general commercial fax instruments. And so you can pass them through these commercial fax instruments, sort out based on things that are happening on the nanovial or things that are happening on the cell, or some combination of both. So how do we make nanovials? So briefly just talk about this, but we have published work on this. We actually use microfluidics to make the nanovials in the first place. We have 2 materials that come in, a PEG, multi-arm PEG that has biotin in some cases, and then a cross-linker that's with a dextran phase. So these 2 materials meet, they form this PEG and dextran droplet. PEG and dextran, if you mix them at the right concentrations, they will actually phase separate to form what's called an aqueous 2-phase system. That means that they're both aqueous phases, but there's a dextran-rich phase and a PEG-rich phase. And so those, once they phase separate, we can then cross-link the PEG-rich phase using UV light in this case, and wash out the dextran-rich phase. And that's how we form the structure of the nanovial. And we can do this at pretty high rates in order to create large quantities of these nanovial suspensions. And these particles can be really, really uniform. So this is an example of showing we're making 50 micron, so opening diameters and 80 micron diameter particles with a coefficient of variation of 1.5%. So really precise structured particles. And this is important if you want to operate with downstream instruments to have this level of precision. And we can make different types of nanovials, right? Nanovials that are suited for smaller cell types like B cells and T cells, or intermediate cell types, some of these cells like HEC or CHO cells, which are producers for producing recombinant proteins or therapeutics. And then also larger cells like mesenchymal stem cells or other stem cells, or even cell clusters. So we can grow those up in the nanovials. I mean, the way to think about this is a nanovial is like a well of a well plate, but this is now can be worked with in the 3-dimensional capacity within a tube, and it can be sorted and moved around between instruments. So as I show here, you can make different sizes of nanovials, and they're, depending on the size, they're compatible with different types of instruments. Standard flow cytometers, large particle sorters, etc., and of course pipettes and microscopes and liquid handlers. So I'm going to go through the steps to use these nanovials. The first step is loading, where we want to basically get cells to be loaded within the cavities of nanovials. The way that we do this is we modify the surfaces of the nanovials in different ways. So I'll show you a few different examples, but generally we can use peptides that bind to integrins like RGD. We can use things like polylysine for some charged interactions, or we can use specific antibodies to bind to specific surface markers on cells as well. So this is just showing you 4 different types of cells. So CHO-DB12, HEC293 or XB-CHO, the type of surface modification that was necessary to get those cells to load well, depended on the cell type. And in particular, XB-CHO cells, which are usually a suspension cultured CHO cell line, were required more additional materials like RGD plus poly-L-lysine or RGD poly-L-lysine plus fibronectin. But generally, you have to tune this for the cell type. But what's also interesting here is these are adherent cells that also we can work with. That's very difficult to do with microfluidic approaches. And then suspension cells like B cells and T cells, what we found is that antibodies to surface proteins work quite well to load those types of cells. So CD45, which is kind of a pan-leukocyte marker, and CD19 is specific to these B cells. So we can load with one or the other, or a combination of those markers. So what is the physical process of loading these nanovials? One approach that we use is actually first settling the nanovials onto a surface. So if you can see this video, what you'll see is that nanovials are settling onto the surface of a well, in this case. And what you notice, if you're looking carefully here, is that almost all of the nanovials have their faces up, faces pointed up. And that's because of the unique shape and center of mass of the nanovial, that they actually settle and will rotate such that their cavity faces are faced up. And that allows you to load cells by seating them on top of the cavity. So that's one method in which we load cells. Yeah, kind of a beautiful ordering. So basically you create your own little array of wells that you can seed cells in, and of course those then can be operated with different instruments downstream. So the process of loading, we started off with a kind of a stochastic process of loading, like you might do if you're doing a limiting dilution cloning or loading into droplets with following some Poisson distribution. So depending on the lambda of your Poisson distribution, you can get different amounts of single cells or doublets or multiplets. In this case, we normally are using, if we want single cells and want to avoid multiple cells, we can use a lambda of around 0.1, and that gives us, maybe 10% or so single cell loaded nanovials. Of course, if we don't care about the multiplets, we can load those at higher concentrations as well. And if we really care about multiplets, then we can load at lower concentrations. But more recently, we've actually been loading in a way that we can beat Poisson statistics. As you can imagine, you can tune the size of the cavity of these nanovials, and we can tune the chemistry. What we found is we can add chemistry just to the internal region of the nanovial, in this case, a gelatin material, so that that only the internal region here is adhesive to cells. And then we can really load lots and lots of cells. And if we size the cavity to about the size of the cell, that allows us to avoid a second cell from entering the cavity, right? Because it's sterically hindered. There's blockage of the cavity by the first cell that's loaded. And this allows us to improve our particle fraction that is single cells. So in this case, we're using a really adherent cell line. If we have a uniform coating all over the nanovials, if we load too many cells, then we'll get some cells kind of sticking on the outsides. However, if we have this localized coating and we overload cells, what we see is that majority, and you can see that the cell size is very close to the cavity size, we get a majority of the cells will have a single cell. And we actually beat Poisson statistics. This is what this is showing, is as we narrow, the particle size decreases to be similar to the cell size, the fraction of particles that have 2 or more cells decreases, beating the Poisson particle fraction that we expect. So this is a way to more deterministically load cells into these nanovials. Okay. The next step after loading nanovials with cells is this optional step of forming tropicals, where you basically compartmentalize the particle with the cells so that you have oil surrounding it, and then you can prevent crosstalk between particles. So this process is pretty simple. You add your nanovials with your oil. You then basically pipette for about a minute up and down, and that creates a dropical emulsion. And this dropical emulsion will form with a very uniform size. And so this is where you can see that. So you can see this uniform size here of droplets. Those are droplets that are templated by the particles, the nanovials. And then you see what we call satellite droplets around. But those satellite droplets don't have a solid phase. They get washed away. And so we don't really care about them in our process. So this, what's really cool, we leveraged a lot of insight from work from like Richard Matthey's lab, about a decade ago, where they were showing that spherical agarose particles could be also emulsified and form these types of emulsions. So how uniform are these dropical emulsions? Well, they're pretty uniform. So this is showing you, the nanovial here labeled in red, the droplet in blue, and the oils in black. And the tropicals that we form have a CV about 4%. So remember, the CV of the particle is about 1.5%. So we're getting a little bit higher CV than the particle size itself. And we have a few number, less than 1% of these tropicals will have these, 2 particles per droplet. So that's pretty good that we get a majority of a single particle per droplet. Okay, so what about assays? So what are the nanovial assays that we can perform using this platform now that we have these tools to load cells and compartmentalize if we need to? And as I mentioned, the first step is really to perform these assays is you need something to perform the analysis with or the sorting if you are doing sorting. And so we've done a lot with flow cytometers. So this is just showing you, what goes in. We have free cells, let's say empty nanovials and nanovials with cells. And what does that look like in a flow cytometry plot? So if those of you are familiar with looking at flow cytometry plots, here's a forward scatter area versus side scatter area plot. What we found is that, by labeling the different populations, what we found is that nanovials generally will be these green and blue populations in scatter, so they'll have high side scatter in this case. And this depends, of course, on the instrument that you're using. But then cell-loaded nanovials will have a higher forward scatter as well. And so you could gate out just by scatter the cell-containing nanovials. Or you can gate out, obviously, if you have floors attached to the cells or the nanovials themselves, you can gate out based off of that and sort populations. But so this is just give you a sense of like what we can start to do with flow, and we'll be using that in some of the other assays that I will discuss later on. Okay, so I want to go back to this slide because I'm going to show assays that connect to various aspects of this of these therapeutic kind of pillars of medicine. The first is identifying producer cells that produce high quantities of antibody, as well as B cells and hybridomas that are producing antigen-specific antibodies. So this is part of the discovery and manufacturing process for monoclonal antibodies. The second I'm going to talk about is T cells that are secreting cytokines. So our ability to analyze live T cells and sort them based on their secretion of cytokines versus surface-bound cytokines, which can be important for cell therapy discovery. And then finally, I'll talk about some results we have for mesenchymal stem cells and sorting and analyzing them based on secretion of growth factors, in particular vascular endothelial growth factor, VEGF, as well as secretion of extracellular vesicles. So this is really great because there's no way now to know what causes the biogenesis of particular cells that produce, that are producing more extracellular vesicles of specific types. And so now we're developing technology that enables you to sort out populations of cells that are secreting high levels or specific types of extracellular vesicles and keep those cells alive. Okay, so I'll talk about these 4 different areas. So the first one I mentioned is producers. These are, in this case, CHO cells, Chinese hamster ovary cells that are producing a monoclonal antibody. And we wanted to select out the populations that are producing high levels of this antibody as a way to select for doing cell line development as one example. And so the process here is we feed the CHO cells onto the cavities. We allow them to, we add a capture antibody for the IgG that they're secreting. We then emulsify. This is important for these types of cells because they produce high levels of antibody, and there's lots of them that are producing antibodies, so we need to prevent the crosstalk. We capture their secreted monoclonal antibody. We then break the emulsion, wash, add secondary reporter antibodies that bind to the IgG, and then we can sort out cells that are producing high levels. So this gives you an example of a spiking experiment. We had non-producing CHO cells and producing ones. And what you see is that when the cells are producing, we accumulate the signal on the nanovial of the secreted antibody, right? So that's, this is, and when you start to sort, you sort out the ones that are secreting. Okay, so let me walk through an assay where we enriched a population of high secreting CHO cells. In this case, they were secreting an anti-IL-8 antibody, and then we regrew that population in order to characterize whether they maintain that phenotype. Okay, so pre-sort, you have cells that are secreting. This is what it looks like. The anti-IgG is in green here. The cell tracker is in purple. So on this is the flow plot here. 488 is the antibody signal, secretion signal. And so we pick this population in the upper right quadrant. We select out the top 20%. We also select out all the producers. It looks good. You get more cells that are secreting when you select out the top 20% or higher secreting. Here it looks like there's lower secretions. We then expanded out the population. And what we found is that we could get up to a 50% increase in the production if we took the top 20% of secretors after 7 days of expanding out that population. So, and that compares to the pre-sort. And if we just select all of the producers, there are some cells that don't produce, that also helped and enriched the productivity of the downstream population. So this is important because you think about it, if you can get a 50% increase in productivity for the same cells over a period of time, that improves your downstream ability to manufacture those monoclonal antibodies at high levels. Okay. We also did this assay on CHO producer cells with, remember, the gelatin PEG particles that were selectively functionalized just to have moieties for binding and capture of secretions just in the cavity. And what we found is that if we just included those capture moieties in the cavity, that prevented crosstalk. And I'll show some data that discusses that. And we didn't have to do the emulsification step. So this protocol is slightly different. We have capture antibody, we capture the secreted antibody without the emulsification step. We label, and then we could sort out the secreting populations from the cell-loaded nanovials. And we could sort 100,000 cells based on secretion within an hour or so. So this is the assay. We have cells that were loaded, they're secreting antibodies. We labeled them without doing the emulsification step. In this case, we tested this crosstalk, right? So we had empty nanovials that we also spiked in, and we looked at how much of the secretion signal was on the empty nanovials. And so these cutoffs here are 1% of the empty nanovials are above this dotted line. So that's a 1% crosstalk. And so what we found is that we could still, 60% or so of our cell-loaded nanovials were above that threshold where we only had 1% kind of crosstalk. And so this is great. We can, so within 30 minutes, you get a great signal. Within an hour, still great. As you go to longer time points, of course, you get more and more secretion, and that crosstalks to the neighboring nanovials. And so that prevents you from having as clean of a measurement. As you can see this here, so for 1 hour pre-sort, you can see cells on some, you can see the secretion signal. It looks like this ring because it's stuck within this cavity. And then once you sort, you can sort out the populations of cells that have the secretion signal. At 4 hours, the signal is increased, and you can see that with the sorted populations as well. So really, this approach allowed you to have minimal crosstalk for up to 2 hours of secretion with this really simplified assay format for producer cells. One thing that we found when we were using these gelatin nanovials to sort these CHO cells is that when we did the sorting process and we compared it to the CHO cells that were in suspension, we actually had an improved viability of cells that were on the nanovials versus the free cells. And so this is likely because first, we're keeping these CHO cells, which like to be in an adherent state throughout the whole process. But second, the nanovial itself can protect the cells encapsulated within them from shear stresses during the sorting process, right? The droplet and air sorters that are common for most FACS systems, there's a lot of shear stress in that process as a droplet kind of impinges, first the generation of the droplet, but then the droplet impinges on the well on which it's being sorted into. We could grow up these cells, out of the nanovials. This is something that people ask a lot, like what happens, can you get them off the nanovials? Well, the simplest way is you just grow them out, right? So they'll start growing off the nanovials onto the plate, and that's an easy way to then expand them. Okay, so one thing that we found is that by using flow cytometers to do this analysis, that actually improved our performance compared to microscopy or microscopic approaches. So this is important if you think about that these generally flow cytometers are like one of the most sensitive instruments that you might have in the lab. And the fact that we're compatible with these instruments allows, really to do the most sensitive types of assays that you would want, using the flow cytometer. So you can see this here, the dynamic range down to about 10,000 molecules per nanovial. And that is improved compared to microscopy. Okay, so I want to then talk about antigen-specific B cells or plasma cells, plasma B cells, and how we can do discovery of antigen-specific monoclonals using this process. So similar to the CHO cell process, we'll seed B cells onto the nanovials. We then add an anti-IgG to capture secreted antibodies from these B cells or plasma cells. We can do that in emulsification if we want. We can then break the emulsion, add fluorescent antigen that we want to have specific antigen-specific monoclonals against, and then we can sort out populations of cells for downstream either regrowth or sequencing to it can be either single cell sequencing or bulk sequencing to identify either repertoires or single matching heavy and light chains for these particular antibodies. So in this case, we've done an experiment where we immunized mice with ovalbumin, and we are identifying ova-specific antibodies. This is just showing you like the control. We took the serum of the ova immunized mice, and yes, the serum, our assay works on the nanovials. We can detect ova-specific antibodies, down to a dilution of 2,000-fold from the serum. Of course, this is not, not all the antibodies in serum are ova-specific, and so the signal here is not that great. But when we're collecting a single plasma B cell that is only producing an ova-specific antibody, then we collect only that antibody on the nanovial, and we'll see a much better signal, as I'll show. And then, of course, we have a control where it's an animal that wasn't immunized, and we see that in that case, there's no signal. Okay, so we did then a whole workflow here where we took CD138 positive cells from the spleens of these ova-immunized mice and also bone marrow in some cases. We could then sort out populations of high secreting cells based on the Cy5 signal for ova. And in that case, we could collect B cells or the CD138 positive antibody secreting cells that had high signal. And so this allows us to then do downstream processing with these cells. We also could do a similar assay. This is just showing a similar assay we did with hybridoma cells that were specific to ovalbumin as well. And then we can, as I mentioned, do downstream assays. So in this case, we're growing up hybridomas after sorting them. And you can see they kind of grow up out of the nanovial. Sometimes they form like these little clusters around the nanovials, but other cells kind of get released out. This is day 3 after sorting. In other cases, we're doing downstream sequencing. Okay, so the next assay I mentioned was cytokine secretion from T cells. So again, the assay format is very similar. You see T cells onto the nanovials. In this case, you have a cytokine capture antibody on the nanovial surface. You let the T cells secrete cytokines. You add a reporter antibody, and then you can sort out based on the amount of cytokine. In this case, you're forming a sandwich, so you have to have two antibodies that bind to the cytokine at different epitopes. And then you can do downstream processing after the sorts. So there's three assays that we've worked on: IL-2, TNF-alpha, interferon gamma. These are kind of the top three cytokines that T cells produce that folks are interested in terms of their function. And what we found here is these are just dilutions that we can basically do an assay for these cytokines on the nanovials, and that flow cytometry is a better readout than microscopy in terms of sensitivity. So here's the assay for TNF-alpha and interferon gamma. So here's kind of the presort images. You have T cells that are activated with PMA inomycin. We then do the staining process. This is looking at TNF-alpha, and this is looking at interferon gamma. So you can see there are some, like this cell here, and then you see the nanovial here has a lot of stain for the interferon gamma signal. This is another cell here. You can see this nanovial has stain for TNF-alpha. We pass these through a flow sorter. You can see here the TNF-alpha signal. Here's the interferon gamma. Sorry, I think this should be interferon gamma. That's incorrect. This is, oh yeah, no, never mind. This is correct. So interferon gamma is on this axis. TNF-alpha is on the y-axis. So here's the interferon gamma signal. Here's the TNF-alpha signal, and you can sort out the high secreting populations. What's really cool here is what we learned, and maybe if you're a T cell biologist or immunologist, you know more about this. But TNF-alpha is an interesting cytokine because there's two forms of it. At least two forms that I'm aware of. There's a soluble form that's secreted, but there's also a membrane-bound form. They have different functions in the cells. So what we can do with this assay format is we can see the secreted form. So here you can see this cell is really not stained brightly, but you can see the nanovial is stained. Here's a cell where both the cell and the nanovial is stained. Here's a cell where there's really not secreted version, but we see the cell stained with TNF-alpha. So you can start to look at combinations of secreted versus membrane-bound signal, and we'll talk more about how you might be able to do that. But yeah, so this is a really interesting format that you really can't do with other approaches. So I'll talk kind of how you can look at secreted versus cell-bound signal here. In this example, we're looking at interferon gamma. So again, presort images. Some cells that are secreting, and you see the nanovials light up. Others where that just the cell is staining. Sometimes this happens if the cell is dead, right? The membrane's permeabilized, and so the stain can get into the cell. What we found is that we can use aspects of the peak in flow cytometry, the fluorescence peak, to sort out subpopulations. So you can imagine that as a nanovial goes through the laser spot of this flow cytometer, that the nanovial itself is much larger than the cell. So as it goes through, you get a wider peak that has a larger area. So like this. A cell itself, if it's stained, that will give you a shorter peak that has a smaller area for the same height. And so what we found is that by looking at this height versus area graph, when we pick out the high area, the ones that the nanovial is staining, versus the low area population, that the high area population corresponds to high staining on the nanovial. So in this case, cells that are producing interferon gamma and that's secreting and binding to the nanovial. But this low area population was that cells themselves are labeled, right? So they're not secreting, but the cells, perhaps because they're permeabilized, are staining brightly for interferon gamma. So this allows you to pull out different subpopulations. Okay, so briefly I'm going to go through a few other approaches, the other assays. So I mentioned extracellular vesicles. So this is a work where we're really trying to come up with a new approach that no one has, which is how do you characterize what makes cells secrete lots of extracellular vesicles, what types of extracellular vesicles they secrete, and how that corresponds to their, what they're producing or what their transcriptomes. And potentially, can we develop, extracellular vesicle producer lines that can help make more EV therapeutics? And then generally characterize single cell heterogeneity in the secretion of extracellular vesicles. It's an unknown, something unknown in the field. So the assay for extracellular vesicles is similar to our other assays in that we have an antibody for CD63, which is expressed on many extracellular vesicles. You could use different antibodies. We then allow the cells to incubate and secrete. In this case, we're looking at 24 hours, a longer time period. And in this case, we looked at mesenchymal stem cells that are secreting lots of EVs. And then we add secondaries, in this case, anti-CD9, in order to bind to the CD9 that's also present on extracellular vesicles. So in this case, we're looking at EVs that are CD63 positive and CD9 positive. So we're finding out subpopulations of cells that are secreting those specific types of EVs. And then we can sort out the high secreting ones and do downstream assays. So this gives you an example of how this looks like. This is before, the sorts. You can see cells that are stained here, and then the EV signal around the nanovial. What we do is we then use that same trick of height versus area to pull out, the cells that are secreting high levels of EVs that bind to the nanovial, and not the cell itself isn't staining. And so this is really great. We're able to get out, both lower concentrations in gate 1, they're secreting lower concentrations of EVs, as well as gate 2, where they're secreting higher concentrations of EVs. This is an ongoing process, and we're now characterizing these cells. And for the first time now, we can answer some questions on like what's affecting the EV production in cells. And then I'll finally end on, because I think we're about 10 minutes. I would like to have some time for questions. I see there's a number in the chat there. It's just another growth factor here that mesenchymal stem cells secrete and is known to be important in their function in some therapeutic applications, which is VEGF. So in this case, we have an antibody against VEGF. We have MSCs that are on the nanovials. They secrete VEGF. We can capture that. We then add anti-VEGF, and then we can sort the VEGF secreting cells. So this is kind of how it looks pre-sort. You can see cell MSCs with the VEGF signal in the nanovial cavity. We can then sort out high VEGF secreting cells. We could sort out subpopulations of those cells for downstream analysis. Okay, so I'm going to wrap up here. So we're really excited by this lab on a particle platform. And I showed you now a number of different applications that we're pursuing, but there's enormous potential to do almost any assay in this type of format. Multiple cells or other different types of assays as well. So our first generation, we're looking at hundreds of thousands of cells that we can analyze per day. We're now, by using smaller nanovials and optimizing our processes, we're in looking at being able to analyze millions of cells, and also cells, both adherent cells. I showed you results for mesenchymal stem cells and CHO cells, as well as suspension cells. And we think there's a lot of advantages for adoption of this type of technology as well, compared to some of the other technologies that are out there. So with that, I'd really like to acknowledge the key folks that worked on this, Joe and Rob, Soyoung, Mani, Felice, and Shreya, James, and Robert, and Sheldon. Right now, so Joe really was one of the leading folks in developing the nanovial technology, which I talked mostly about, and he's now leading a company that I co-founded and I have a conflict with, Partillion Bioscience. Partillion is hiring now, so if you're interested in all the work that we're doing here and what's next with lab on a particle technology, I encourage you to look at the career opportunities at Partillion as well. So with that, I think I will end and happy to answer some questions.
Thank you for this fantastic and inspiring talk, Dino. Many questions came in, so let me start with the chat box. How do you control the symmetry of nanovials during fabrication? How to ensure the uniform functionalization inside the vial cavity or density of available functional groups for receptors?
Right. Yeah. So the process of manufacturing is using this aqueous two-phase system, right? So that system is basically a surface minimizing system. It's an energy minimization process. So the minimum energy is achieved when you have a symmetric droplet within a droplet. And so that's why you have a symmetric nanovial structure.
How long can your culture grow in the nanovials tropicals? What are some strategies if you need longer culture time for the single cell assay?
Yeah, you can grow, as long as you want. Of course, as cells grow and divide, they'll fill up the cavity. And at some point, they'll overflow the cavity. So that's the limitation, is the size of the nanovial that you use for how long you can grow. If your cells don't divide rapidly, then you can grow longer, right?
Thank you. What is the radius of the nanotropicals and what are they made of?
Sorry, can you repeat the question?
Yeah, what is the radius of the nanotropicals and what are they made of?
Yeah, so we have a number of different sizes that we've made. They're made out of PEG, polyethylene glycol, mostly. We make sizes from 30 micrometers in diameter or so up to 100 micrometers in diameter or so, or even larger in some cases.
Thanks. How well can you wash out the dextran during the nanovial fabrication?
Yeah, the dextran washes out completely.
Nice. What is the smallest nanovial or tropical you can create?
That depends on the ability to make microfluidic droplets at that. So if you can make droplets that are smaller, you can make these smaller. We mostly want to operate on cells. So in that case, you wouldn't want to go smaller than the size of a single cell, right?
I see. Are there other type of hydrogels that could be used instead of PEG to fabricate the nanovials?
Yeah, as long, so the process that we use is with aqueous two-phase system. So if you could, as long as you can, there's also other ways that you can make this type of shape, but yeah, if you could find another ATPS system that behaves like this, that would be probably the easiest way, but there are definitely other ways to make this shape.
Thank you. Is it possible to trap nanovials such as with optical tweezers?
Should be. Yeah, the hydrogel will have a different index of refraction than the surrounding water. So in that case, once you have an index difference, then optical tweezers should work.
One person was asking, can you make a biological switch from the nanovials? Bistable switches based on like a toggle switch.
You could probably test those switches using the nanovial. So like the switches you usually are including in cells, right? So you could test the switch on the cell that's on the nanovial and doing something based off of that switch.
Thank you. Thank you. Would you please explain what is the benefits of this method in comparison with microencapsulation, such as liposomal iron, zinc, etc., based on free cells and cell-loaded nanovials?
The liposomal encapsulation, is that what the question is?
What is the benefit of this method in comparison with microencapsulation, with liposomal iron, zinc, etc.?
I'm not sure I understand that completely, but so the process of using the nanovials is you don't need a microfluidic device. The people that use it, they just they load cells and things onto them. So one of the key advantages is you don't need any microfluidics to use nanovials. Of course, you can also use them with flow cytometers, right? So and they have a solid phase in which you can perform assays, which you might not be able to with a liposome approach. That I'm not sure all the details of what you're referring to, but.
Well, I think I got it, thanks. Did you compare epithelial cells and blood cells to assess the effects of shear stress in FACS and the benefits from encapsulations? It might be cell dependent.
Yeah, it certainly could be cell dependent. We haven't done a complete sweep of all different cell types, but I imagine, that it's known that there's viability challenges with FACS, especially for adherent cells. And so I imagine that you'll get improvements using the nanovials for a majority of cell types, but we haven't done those tests.
Thank you. Have you explored tropicals made of other materials such as silica?
We haven't used other materials in our experiments so far.
I see. For the seeding stage, what is the percentage of cells that are successfully seeded into the nanovials?
Yeah, that's a good question. It depends on the approach that you're using to seed. If you're like overwhelming with lots of cells, then it can be a lower percentage. But if you're kind of seeding a lower number of cells when you have a large number of nanovials, you can get much higher percentage of seeding.
Thank you. How do you control the deposition of gelatin to cover only inside the vial?
Yeah, so the gelatin peg is an aqueous two-phase system that forms, and so gelatin will localize into the gelatin-rich phase, and so that leads to gelatin localization in the inside of the nanovial.
I see. In the process of capturing cells into these particles via antibodies, how do you make sure that antibodies are bonded only inside the cavity, not on the surface?
Yeah, it turns out that it doesn't matter too much. So you can have antibodies on the surface as well. But because the surface is not protected from shear stress, cells that attach there will get sheared off. And so only cells in the cavity, those cells become more stably trapped. Also, we have that gelatin approach that I mentioned, right? In that gelatin approach, then you have local antibody functionalization only in the cavity.
How do you maintain the viability of cells inside the cavity?
The cavity is open to media, right? So there's no challenge for maintaining viability.
I see. Can you give a brief comparison with droplet microfluidics, such as pros and cons over droplet assay?
Sure, yeah. So a droplet assay, you have to have a microfluidic device to do that. You don't have necessarily a solid phase to do any assays, and you can't easily do solution exchange to do complex assays like the immunoassays that I mentioned, where you're changing, adding a secondary antibody. And then you need a specialized sorter in order to sort your droplets. And usually that's going to be less available or less sensitive than a flow cytometer.
Thanks. Have you tried, or is it possible to apply this technology to a heterogeneous population of multiple cell types to test for multiple molecules simultaneously?
Certainly. It's possible. Yeah, we've looked at, we're looking at rare cells. The applications I showed you are rare cells where there's heterogeneous populations of plasma B cells, for example.
I see. Is there any issue with burst of nanovials running through the fax machine due to the high pressure?
No.
I see. What is the efficacy of the nanoparticles to catch one single cell?
Not sure I understand what that means, but I did talk about the ability to control the Poisson loading beyond Poisson loading. So what I showed was that you can actually improve the loading to have only single cells beyond kind of the random process because of the sizing the nanovial cavity to the size of the cell.
I see. Can you use these nanovials for drug delivery?
Potentially, I'm not sure, what application you're thinking of, but potentially.
I see. What is the future perspective for using these nanoplates? What are the next steps to improve?
Yeah, so I think, we showed a bunch of applications, and so this is a interesting kind of general approach that if you think about generally, how do you do biology, right? How do you do single cell assays? We see this as a platform on which you might do any type of single cell assay. And so we focused initially on assays that we know there's a lot of interest in, because, as I mentioned, these are connected to these three pillars of medicine where we're developing new therapeutics. And so that's kind of the focus initially, but you can imagine applying this to other types of things that cells secrete or release beyond, antibodies, cytokines, extracellular vesicles. You can look at other types of functions, cell killing or cell-cell interactions, these types of things.
One final question. There are some limitations, disappeared. There are some limitations of precisely measuring some cytokines or other molecules such as VEGF from MHCs. Could this method, lab on a particle, address this issue? And what are the limitations of this method currently it needs to overcome?
Yeah, so I mean, I think the key thing here is that we are overcoming limitations, right? Because there's not, so maybe I didn't clarify this part. You know, antibodies produced by CHO cells, they produce at huge rates. You know, it might be 1,000 per second. Cytokines from T cells, it might be like 20 per second. VEGF might be like 5 to 1 molecule per second that's being produced. So we were able to kind of span that different range of secreted molecules because of the small volumes. And the fact that the cell is localized within that cavity allows you to concentrate the secreted signal so that you can drive the reaction onto the surface. And that gives you much higher sensitivity to really small amounts of secretion.
I see. Have you explored other ratios of dextran and PEG to change the geometries?
Yes, we have. You can look at our bioRxiv paper, that has more details about that.
Awesome. Awesome. Well, thank you for the great talk. That's all the questions I have, Dino. Thank you for your time. Appreciate it.
Thanks, Mehmet. Thanks, everyone. Appreciate all the comments and questions, and happy to take more questions by email if of interest.
Thank you, Dino.
Thanks, Ali. Take care.